Welcome to my channel XY Biology Academy. Let's understand the working of the fluorescence microscope using the ray diagram. The process begins with a Mercury Arc Lamp, which serves as the light source. It produces an intense beam of ultraviolet or blue light that provides the energy needed to excite fluorescent molecules. The light then passes through the exciter filter. As its name suggests, this filter selects only the wavelengths capable of exciting the fluorochromes while blocking all unnecessary wavelengths. The selected light next reaches the dichromatic mirror. The term dichromatic simply means that it works with two different wavelength ranges. It reflects the short wavelength excitation light downward while allowing longer wavelength fluorescent light to pass through. The reflected excitation light then travels through the objective lens and is focused onto the specimen. In fluorescence microscopy, the objective lens performs a dual function. It focuses the excitation light onto the specimen and later collects the emitted fluorescent light. The specimen is stained with fluorochromes, which are fluorescent dyes that bind to specific cellular components. When the excitation light strikes these fluorochromes, they absorb the energy, become excited, and almost immediately emit visible fluorescent light with a longer wavelength. This emitted fluorescent light travels back through the same objective lens toward the dichromatic mirror. Since its wavelength is now longer than the excitation light, it passes through the mirror instead of being reflected. The light then passes through the barrier filter. As the name indicates, this filter acts as a barrier by blocking any remaining excitation light while allowing only the emitted fluorescent light to pass. Finally, the purified fluorescent light reaches the eyepiece or camera detector, producing a bright, high-contrast image in which only the fluorescently labeled structures are visible against a dark background. Let's summarize the complete light pathway. The Mercury Arc Lamp generates ultraviolet or blue light. The exciter filter selects the required excitation wavelength. The dichromatic mirror reflects the excitation light toward the specimen. The objective lens focuses the light onto the fluorochrome-stained specimen. The fluorochromes absorb the energy and emit fluorescent light of a longer wavelength. The emitted light is collected by the objective lens, passes through the dichromatic mirror, then through the barrier filter, and finally reaches the eyepiece or detector to form the image. One of the greatest advantages of fluorescence microscopy is that it allows scientists to visualize specific parts of a cell instead of the entire cell. This is achieved by using different fluorochromes or fluorescent antibodies that bind only to particular cellular structures. Thanks for watching. Like this video and subscribe to my channel.